Good morning, class. Good morning, class.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Can somebody confirm?
Yes.
Can somebody confirm if the class is doing good morning, class?
Good morning, sir.
Good morning, sir.
Can you please confirm? Can you please confirm if I'm on? If I'm being if I'm on live on YouTube, confirm.
Yes, you are.
Can I see your screen?
If the class is doing good morning, class.
Good morning, sir.
Good morning, sir.
Can you please confirm? Can you please confirm if I'm on? If I'm being if I'm on live on YouTube, confirm.
Yes, sir, you are there. Yes, you are.
Yes, sir. You are, sir. We can see your screen, sir.
Good morning, class. Hello, class. Am I on with you?
Yes, sir.
Yes, sir.
Yes, sir.
Now I think it's okay now. That was a throwback. Is it from alert now?
Yes, sir.
Okay, and is my screen visible now?
Yes.
Now let me confirm again. Is my slide still being shared?
Yes, sir.
Can you confirm if my class is still being shared? I'm having some issues from this time. Is somebody manipulating my? Sorry, is somebody? Hello, class. Am I saying on
Hello,
with
sir.
you, please?
Yes, sir.
Hello?
We can hear you, sir. We can hear you, sir.
Okay, somebody is doing something over there, and it's affecting me in this place. Somebody is
It should be-
pressing the transcribe and is as engaged transcription. It's really disturbing. That person should please stop. If you are the person, please stop it. Please, when you are signing your name, before then, please log in with your proper name. No acronym, no AKA, your proper name, your department or program, and possibly your matric number. That helps us in taking attendance. So this is very important. Now, in this part of the course, we'll be talking about ecological sampling method. And we will look at what it entails to engage in ecological sampling, the step-by-step and another thing to do, how we collect our samples, the methods of sample collection, data analysis, and other things. So that is what we are basically going to look at in this part of the course. Now,
Sir, please, we can't hear you again.
is this okay now, please? Can you confirm?
Yes, sir.
Am I on with you now? Please, am I on with you now?
Yes, sir, we can hear you now.
Okay,
Yes,
good.
sir.
Okay, good. Thank you very much. I was having issues from this end. And my slide is showing now. Is it? Am I sharing my slide now?
Yes, sir.
Yes, sir.
Okay, good. So we are talking about ecological sampling methods. Now, in ecological sampling methods, this covers a wide range of techniques required to study a range of organisms from the field. Now, our sampling location could be in the open field. It could be terrestrial habitats, which I said open field. It could even be a forest. It could be a wetland. Is that okay? It could even be an aquatic habitat. So you may want to sample from any of these habitats. So it covers, you may be interested in the study of a single organism, species of organisms. You could be interested in the study of the population. You could be interested in the study of the community. When we say community, we are referring to all the living organisms of a particular habitat, including the plants, the animals, and even microorganisms. So depending upon the design of our study, ecological methods cover all these. So and different techniques are available to be deployed to sample appropriate study sites.
Sir, we can't hear you.
Sir, we can't hear you.
Said, sir.
Is this okay now?
Yes, sir.
Now, at what point did you lose my audio?
Like
So that
10
I can go
minutes
back.
ago.
10 minutes ago? Wow.
Yes, sir. did not hear your explanation on the second slide.
Sir, we are not hearing you.
Now, I think I'm audible now.
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So I'll go back to what I've said. I was trying to say that before you engage in ecological studies, whether field studies, whether aquatic studies, that there are defined steps to take. And the first step the first thing to do is to formulate the aim and the objectives. After then, you go to the field and take your samples. After you are taking samples, you are taking data from the field. You come back to the lab and analyze the data you have collected from the field. From that, you can make deductions and even go ahead to formulate other things and then arrive at a conclusion. And you have your result. So the 3 steps basically are: define the aim and the objective. 2, you go to the field for sample collection. 3, you come back to the lab to analyze the data you have collected from the field. So let us see each of these steps. What do we mean by the aim? The aim of a study is the goal of that study. What is the aim? That is the overall intention. What is the goal of the study? And then you also define the objective. Now, what are the differences between the aim and the objectives? Though they work together, but they mean different things. The aim is usually broad. That's the goal, usually broad. What is the purpose? Why engaging in this study? You know, it's a broad focus, very broad. And then after you have defined the aim, probably your aim could be a field study. Okay? The aim could be to determine the population size of a particular species in this habitat. And what do we mean? The species, a particular species in a particular habitat, that refers to a population. Don't forget, in your ecological study that you did, you were taught the definition of some concepts, including species, population, community, ecosystem, and so on and so forth. You were taught that species are members of the same organisms that can interbreed freely. That's a species. What defines a species is the ability to interbreed freely. 1. Now, a particular species of organisms living together in a particular habitat, that is a population. That is to say, the population consists of organisms of the same species that live together in a particular habitat at a given time. So when we say that you could be interested in the study of a particular species of a particular study site, species members of that habitat, they make up the population. Please, if you're forgetting that one, remember now. And when we say community, all the living organisms of a particular habitat or ecosystem, they make up the community, or better still, biological community. So that's the thing. Now, so the aim would be to determine the population size of the tilapia zile in a particular pond. That's the aim. It is broad. Then, what are the objectives? When we say objectives, objectives are the steps to be taken to achieve the aim. Okay, now you want to you have a particular pond. You want to determine the population size of a species, the tilapia zile. What are the steps that you can take? So that is what is referred to as objective. It could be the first thing I will do is to determine the appropriate method that I will use. That's objective 1. Objective 2 would be, okay, I will now go ahead to employ the technique that I've chosen for this study. For instance, now, you may want to do capture the capture. So, step 3, I will visit the site and make a catch of tilapia zile. I will count and record. Objective 4, I will make indelible marks on the captured tilapia zile and release back to the population and go away. Objective 5, after 2 weeks, I will go back and make another capture. Objective 6, I will now look at the ones I've caught for any one of those caught that have marks. So that is what is meant by objective. The aim is the goal. The aim is broad. The objectives are specific. Objectives are the steps to be taken to achieve the aim that you have set. I hope that is clear enough. Now, after that, what do you do next? You now go ahead to do your sampling. Now, the next question to ask is that, how do we sample? How do I sample? There are different ways or methods that you can deploy for your sampling. Depending upon the type of habitat, depending upon many factors, how large the study plot is, resources available, the time available, all these, you will take them into consideration and arrive at a particular method or techniques that we will deploy for sampling. And of course, the best method to get the appropriate number of any species is to do what? Is to do counting. A sort of what? A sort of play. Another person, Joy Oluwakanyisola, can you please mute yourself? If anybody saw my class, I will throw that person out. Please. So we go ahead now. So let us now see the different sampling techniques. Okay, so like I said earlier, the best approach to get the most reliable method is to do counting one by one. However, it is for many reasons, it is not possible to want to do edge-to-edge sensors counting for many reasons. For many reasons. Usually the sampling plot or location is wide, limited time, limited resources. Now, if the direct counting or sensor is not possible, what are the things to do? That is the essence of doing sampling. Now, when we sample, sampling is taking a portion of the study habitat that is representative of the sampling location. Is that okay? Now, basically, there are three major types of sampling. Random sampling, systematic sampling, stratified sampling. Each of these sampling methods have their advantages. They also have their disadvantages. They have their peculiarities for each of these sampling methods. So we are going to see each of them and when that will be used and when they should not be used. Let us look at random sampling. When we say random sampling is a sampling method in which each location of the sample field stands equal chances of being selected. And the selection of a particular unit does not influence or interfere with the chances of selecting any other. Random sampling. In mathematical terms, we say that the locations are what? Independent and analytically distributed. The sampling of a location, the chances of every point, every location of the sampling field stands equal chance of being selected. And the selection of a particular plot does not interfere with any other plot being chosen for selection. So that is random sampling. Now, what are the equipments or techniques to be used in random sampling? That's the next question to ask. Generally, there are many random sampling methods or techniques that you can use. However, for terrestrial ecological studies, two major instruments or methods that are used are, number one, the quadrat sampling. Number two, transect sampling. Sorry, yes, I'm right. Number one, quadrat. Number two, transect. We are going to see each of these. So before then, for any of these conditions, like I said earlier on, there are conditions to be fulfilled. when do we use random sampling? Random sampling is used when the area of the other study is large. That is one. It is used when the area of that study is fairly uniform. The organisms are fairly uniformly distributed. Number 3, resources and time that are available are also limited. So if those conditions are met, then you can deploy random sampling. However, we must ensure that we take a large number of samples. The more the number of samples taken, the more representative or reliable the results become. So that is the conditions for random sampling. So now back to what we have worked before. 2 common techniques. Lawal, light chat. Please don't disturb me again. If you do it again, I will throw you out. Can you mute yourself, please? Lawal, light chat. Olawunmi. I saw some people are disturbing my class. This is not good enough. Hello, class. Am I back now? Can you confirm?
Yes,
Yes, we
sir.
can hear you, sir.
I want to appeal to you, for goodness sake. Please, if I don't ask you to unmute, please don't unmute. Please, I beg of you, please don't disrupt the class again. Please, for goodness sake, please. Let's have a smooth class, please.
Sir, please, I can't hear anything. Or is it only me?
Well, Ajibola, I'm very audible enough. You check from your end. Check from your end. At a point, somebody disturbed the class. So I went off, I pulled that person out, and I came back. And I know that now I am on. Class members, can you omit and confirm that? Am I on with you now?
Yes, sir.
Okay, good.
Yes,
Yes, we
sir.
can hear you, sir.
Okay, thank you. So, what are the tools for random sampling? Two common ones are the quadrat sampling and the grid sampling. Quadrat and grid. Now let us see the quadrat. What is a quadrat? The quadrat is a square or rectangular frame made of wood or metal. Usually made of usually a square, and most often made of wood because wood is easier to construct. It is usually used for something for fish study. It is thrown severally at different points of the fish. So these are quadrats. This is a quadrat by the left, another quadrat. Within the quadrat, you can have smaller, you can divide into smaller plots. Is that okay? If you can see in this space now, the vegetation is uniformly is uniform. In this field, you will notice that the vegetation is appearing in patches. So in this place, it is good that if the plots, if each plot is divided into smaller units, as in this case, what we are saying is this: your quadrat will be divided into subunits, some plots within it. If you see, okay, so that's a quadrat. What do? You throw it to different directions, or you carry it, you lay it. It's either you throw or you lay. And in some cases, now you can only throw where the study field is mainly herbaceous or grassland. But where there are trees, it may not be easy to do throwing. Even at that, it may not be easy to be laying the quadrat. What can you do? You can use tape to draw out the area of the quadrat. So like I said earlier on, it's still saying the same thing here. The quadrat is thrown severally at random within the study site. This is done many times. The more the number of throws, the more reliable the results. Again, this slide is talking about the conditions for using a quadrat. The field under study is uniform, like a grassland. When the organisms under study are plants or sessile terrestrial organisms, sessile are dormant or slow-moving organisms, or that immobile. They don't move. Even if they move, the movement is very insignificant and limited. So in such cases, scenario, you can deploy the quadrat. Now, if your species under study is a moving animal a fast walking animal. Is this appropriate to use the quadrat? No, it is not appropriate. So after you have collected, thrown your after you have thrown your quadrat several times, for each throw, you go to the plot. You count the number of that species. If you are interested in Tradas cucumber or that quadrat, Tradax in each throw, you count it and then you record. From your data, Joseph, can you please meet yourself, please?
Oh, sorry. I don't know what it is.
So from your data you have collected, you can calculate many things from your data. Frequency, total number of the species, the density of the species, the dispersion of the species under study. So you can calculate all those things and even more from the data you have collected. Don't forget what you did in general ecology. And that is where this, if you go back to your manual and your notes, all these things are there. You did them first semester when you are studying general ecology. So population frequency is calculated as number of quadrats that the species occurred over total number of quadrats thrown. You can calculate relative population frequency, individual population frequency over total population frequency. You can calculate population size as indicated in this equation. You can also calculate total density. Please go back to your general ecology, the particles you did, go back to your notes, go back to your manual, go back and check the answers you wrote for the practicals. I don't know if you even wrote it for the exams. So this one, they come handy here. So we just made reference to it. Let's go on to see. Let's see, go on now. Now, what are okay, when we talk about dispersion measures the pattern of distribution of that organism, usually plants. Whether they are distributed.
Are you serious?
Zebra. Olagbaye. Why now? Okay. Now, what are the limitations to the use of the quadrat? One major disadvantage or limitation of the quadrat is that it is prone to personal bias. It's a major disadvantage of the quadrat. It is prone to personal bias. That's number one. Number two is that there's also limitation of size. There's a limit to the size of the quadrat you can be carrying about. If it is too large, it becomes difficult to carry about and to work with. So that's a challenge. Three, you can only throw quadrat when the study size is uniform, like grassland. Where there are trees, where trees and shrubs predominate, quadrat may not be thrown about, but then you may find a way around it mark out the area under study, and then you take as a quadrant, as a throw. So which again is a limitation because you have to be mapping out, maybe with a rope, to map out to the equivalent to the, you map out to the equivalent of size of the quadrant, and then you go ahead another point. So it's also time consuming. That's 3. 4 is that species that are rare, that are not common, that are rare, may either be underestimated or overestimated. So all these bias are associated with the quadrant. The major one, number 1, is prone to personal bias. Number 2, there is limitation of size. You can't be carrying a quadrant that is too large. Number 3 is that if the area under study is not uniform, it's also a challenge. Number 4 is that some species that are rare, not commonly occurring, may be either underestimated or overestimated. All these bias are associated with the quadrant sampling. So what then can we do? Yes, we can begin to look at another alternatives, especially when the challenges are pronounced. So to mitigate or to reduce personal bias, we can use grid sampling. What is a grid sampling? In the grid sampling, the first thing you do after you have identified your study site, you map it out. You map it out, and then you divide it into grids. Now, you will now generate some information or data from the computer, which will guide you in choosing which grid to sample. So 1, the area under study is mapped and numbered, and then placed over the map. So you map it out, you number it, you now lay the grid. I mean you now lay it off. You lay the map over the study site, and then you use a random number table, usually generated from the computer. That will guide you in selecting which of the squares to sample from. Let us see this. Now, for instance, this is the map. Can you see my mouse? Can you respond? If you can see it.
Yes,
Can you see as
sir.
I trace? Okay, good. Thank you very much. So this is the map of the study area. This is the map. This is the map. This is the map of the study area. Now you have divided them into grids. Excuse me, please. Let me respond to this. I'll go back now. Hello, class. Am I back with you?
Yes, sir.
Okay, good.
Yes, sir.
Okay, good. So these are the grids. 1, 2, 3. So this is the map that are divided into grids. Good. So now let us now see. So now you have generated a random table number. Can you see? Where is it again? Okay. This is the sampling table. Random table number. This is generated from the computer. And this is your grid. Now, how do you now do? What do you do? How do you now determine which of these grid to sample from? So this is the sampling. Now, the sampling table number will have guided you which number to start from. Now, for instance, now, if you are starting from 40, now this is the mouse. My mouse is showing 40. If the number says that sample, you may want to sample, you sample right, left, you sample right, you sample up, you sample below. So number 1 is 40, isn't it? If number 1 is 40, what do? You go to look at number 1, number 40, and sample. Let us see it. Is there 40? Is there 40 in this plot? Can you respond?
No, sir.
No. Then what do we now do? Since 40 is not in our plot, we will skip it and go ahead. Now don't forget, here's 40 again. Where am I again? Yeah, okay. Don't forget, we are going down first. So 40, we skip it. What's the next number down? 15. Let's now go back. Where is 15 in the map? Well, okay, now we now go ahead to we now sample from this plot 15. After that, don't forget we are going below. Okay. What comes next? 50. So let's go back to the map. 50 is not there. Again, we skip. So we are taking 5 up, 5 down, 5 this way. So 69 is not there. Then lastly, this is 5. Number 5. Number 5 is 6. We go again to 6. That's it. So 6, 6, 6, 6, 6, 6, 6, 6. Now, we sample this point. Don't forget, the first one we sampled was 15. The second one was 6. Then you go back to 40. Since we're looking at 5 up, 5 down. This is okay. Now up. What number is the first number up? It's 11. You go back. We sample from here next. Good. Now you go back. The next number is 2. You go back. We sample from 2 here. Good. You go back. That's what we have up. And then from this 40 again, we go right. We go left. And then you are through. That's okay. You now go to the next point until you are through with sampling. So that is the simplest way to explain grid sampling. Okay, so this lets explain what we just said, although using different numbers. That's okay. Looking at the top, this one starting from 20, you move down 74. So this one is just explaining what I said earlier. It's repeating what I said earlier. So now let's see. Now, in some instances, it will not be possible to use the quadrat. It will not be too visible again to use the grid. Then you may deploy random walk. Again, random walk is determined by some computer-generated number. So if the first set of number is saying 1735. So 1735. 17 indicates 17 indicates the degree. So 17 degree. So if it is 360, 17, where will 17 be? Let me see. 0 to 360. So what is 17? 17 degree will be a little bit. Where will it be? 17 will be at where? 17 will be at something like 2 o'clock. Am I right? What will 0 be? 0 will be in the south. I mean the north. Straight north. Isn't it? 45 would be what? Right angle. Right angle would be what? Will be 3 o'clock. 180 will be 6 o'clock. Do you get it? Do you get it now? And so on and so forth. 270 will be what?
Yes, sir.
270 will be
9.
like is it 9? Okay, something like 9 or 10, between 9 and 10. Am I right? Something like that. So now the first was what? 17. What follows 17? I mentioned the number. I just at random. What was the number I mentioned? Okay, if it is 1754, 17 refers to number of the degree. 54 refers to number of steps that you will count. That is, you move one step, 1, 2, 3, 4 until 54. So that's the idea of random walk. And this is what this slide is trying to explain. Let's go ahead now. So that is about random sampling. Don't forget, generally speaking, I said we have how many sampling methods? Number 1 was what? Random sampling. Am I right?
Yes, sir.
Number 2 was what? Systematic sampling. Am I right? Yes.
Yes,
Number 3 was stratified sampling. Yes, good. Now we are talking about random sampling. We said, what are the instruments that could be used for random sampling? we said we have the quadrat, we have the grid. Where those two don't work, you may use random walk. We talk about the peculiarities of each one. We look at advantages and the shortcoming challenges associated with especially the quadrat sampling. And then we look at what the grid is, how to use the grid, and then where and how to use random walk. So that is that with random sampling. The next method is systematic sampling. One instrument that is used in systematic sampling is the transect. What is a transect? A transect is just a row or a tape that is marked at fixed intervals. That's a transect. So transect is used in systematic sampling. Systematic sampling is of two broad types. Transect sampling. Let's see. Okay. So this slide is telling us about systematic sampling taken at fixed intervals. And when do we use it? When when we use random sampling. Random sampling is used when the area that other studies is uniform. But when it is not uniform, when it is varied, at times, when the location is varied, it's not uniform. It's varied. It's not too large. Maybe you have some either it's uniform with plant, some is marshy, aquatic, you understand? Or there is a slope or something. So you decide to use systematic sampling. In such situations, it won't be appropriate to use quadrat sampling or degree sampling. So you now deploy systematic. Systematic sampling is the use of transect. So, and these are the conditions. The area under study is varied, and it is not too large. And then the time limitation is not there. Now, this, you can see this study site. You can see that it is varied. You can see aquatic area. You can see vegetation, and you know. So this, how many transects are here? Two. One is yellow, the other is red. This is one. This is one. So that's just a transect. Now, transect sampling could be. Okay, you can see this one too. You can see that there's a gradient in this sampling site. There's a slope, there's a hill, there's a descent. You cannot be using quadrat or grid sampling in this case. The best thing to do is to deploy transect sampling. Now there are two methods of transect. Number one is the line transect method. Number two is the bed transect method. What is the line transect method? In line transect method, this is the line. You use the transect. This can be continuous or interrupted. Continuous is that when you lay the transect, don't forget that the species of plants You have identified it. So any species of that plant that the line or the rope is touching is counted and recorded. This other one, system beds, these beds you are looking at consists of the line and the quadrat. We see that very shortly now. So generally speaking, there are two types of transect: line transect method and bed transect method. So I said this earlier. The line is laid across. Line transect could be continuous. It could be systematic sampling. Now I will go now. Okay, good now. Good. Now look at this researchers. What does this table represent? This table represents the transect. If these quadrats were not there, what do we call that? Somebody tell us. What do we call that? We can see these students using the tape rule. This tape rule corresponds to the transect. You can also see them laying quadrats across it. If these quadrats were not there, what do we call it? Anybody? Okay, let's go back.
Belt transects.
No. I said if the quadrats were not there.
Line transect.
Line transects.
Continuous line sampling. Line transect. You are assigned. Continuous line transect. Sorry, line transect. Line transect could be continuous or systematic. If it is continuous sampling, what is that? Any species that the rope touches is counted. If it is systematic line transect, what does that mean? It means that the transect is marked at different intervals. So you could begin to count maybe from 1 cm, 10 cm, 20 cm, 30 cm. You get it? That is systematic line transect. However, if you don't use interval, you just count every one of them along the line, that is continuous. But if you count at given intervals, that is systematic line transect. That is one part. The next part is that it could be bed sampling. In bed sampling, you lay the transect, you also support with quadrats. So that is That is a bed transect sampling. That one too is of two types. Don't forget, line transect was of two types: continuous line transect, systematic line transect. Then you have bed sampling, where you deploy quadrant with the line. That one too is of two types. It's of two types. These quadrant now, they could be spaced out at intervals, or they could be placed end to end. That is adjacent to each other. If it is placed end to end, no space in between, we call that bed transect. If there are spaces between each quadrant, between the quadrants, that is interrupted transect. Now, in this case that I'm looking at, what type of transect are they deploying? Are they using? Somebody tell us. Is it bed transect or interrupted transect? I'm waiting for a response. If you are there, can you unmute and let me hear you talk? You can see in the class. Can you let me hear your voice?
Yes, sir. Yes, sir.
The question I asked, is this clear to you? I'll ask a question. These students now carrying out their study, what type of bed sampling or bed method are they using? Is it ordinary bed transect or interrupted transect?
Interrupted transect.
Interrupted transect. Why? This is because there are spaces between the transect. If the spaces were closed up and transect is placed one next to the other, end to end, what do we call that? Somebody
Belt transect.
bed transect. So that is that for bed transect. Don't forget, all this is coming under what? Systematic sampling. The first thing we talked about was random sampling, followed by systematic sampling. That is what we talk about. So we are talking about random sampling. We talk about systematic sampling. Now we say there are three types of sampling. Isn't it? Random. Am I right? Systematic. What is the third one? Stratified.
Stratified.
Now, what instrument do we usually use for random sampling? Somebody on it can tell us.
Quadrant.
Quadrant 1.
Grid.
Grid 2. What else can, if it's not possible to use quadrant and grid, what can we use?
Working method.
We can use random walk. I mean, random walk, yes. Random walk. So that is random. Now, for systematic sampling. What instrument do we usually use? Somebody on me can tell us. Don't forget the instrument we use for, okay, we use transect, yes, transect. And how many ways can we use the transect?
2 ways.
2 ways, yes. It's either we use the only,
Your
only-
mom, please.
yes.
I can't
It's
remember.
only- it's only we use, it's either we use only transect,
I don't remember.
or we support by
I'm not allowed to try, but
fine. Why now? Don't disturb us again. So for systematic, we use transect. It's either we use transect alone. I will throw this to out. Why now? So it's either we use only transect or we combine with quadrant. Good. So let us briefly see certified sampling. There are some locations. There are some locations that you may not be able to use systematic sampling or random sampling. You understand? We use stratified sampling. I will not, I can't allow this again. What is this again? Why are you doing this? I will throw you out. Now, there are some study locations that are very varied. You understand? That, so you use stratified. Stratified sampling takes into account different area or strata. You understand? That is why the name is coming from, strata. Different strata. Now, look at this. Look at the distribution of organisms in this study site. Is it too good? If you use the quadrant in this place, are we going to get reliable results? No, we wouldn't. We will miss out. We may underestimate this green area, or we overestimate it. We may underestimate this. What color is this? This pinkish area. You can see, the organisms are very different. Some occur in patches. So we cannot deploy either random sampling in this location, or even deploy systematic sampling. What we can use is a method that will take into account every location, every strata. Now, not every location strata, we stratify it. These green locations is a strata. This pinkish location is a strata. This sparsely populated location is another strata. So we have to ensure that we sample to cut across each of these strata. So that is what it means to use stratified sampling. This method takes into account different areas of strata that are identifiable within the study habitat. And then each of these strata is sampled differently from the main part of the habitat. For the other part of the habitat, you might have used, for instance, now, this could be just a part of a study plot. Just a part. So you might have used random sampling for the other parts or systematic sampling. But this area, this location of the feed, you now deploy certified sampling. So that is it for random sampling, systematic, and certified. Now, all we have been talking about since we started this class is focused on terrestrial habitats. You know, we all focus on terrestrial habitats. Now, supposing the study site is aquatic habitat, is it possible to use further sampling? No. Is it possible to use transient sampling? No. Is it possible to use random and stratified together? No. Is it possible to use systematic and stratified together? No. So, in aquatic, for aquatic habitat, there are different methods. There are different methods entirely that are used. So all we have been saying is about sampling in a terrestrial habitat. The next thing to do now is to look at sampling in aquatic habitats. So far, let's ask questions. If there are questions before we go ahead, before we go on. If there are questions, please feel free to ask now before we go on. Yes. If you're in the class, can you omit and let me hear you talk? If you're in the class, you're in the class.
Yes, sir.
Yes,
Only
sir.
one person is talk. Yes.
Yes,
Yeah.
sir.
Okay.
Yes, sir.
Yeah. Now, if there are questions, let me hear you ask your questions now before we go on. Now, let me ask you now. Can we go on now? Can we go on?
Yes, sir.
Yes, sir.
Now, yeah, okay, good, very good. Thank you very much. So let us now go on to see aquatic sampling. I will soon be through. I will leave this class before 12. The class should end by 12, but I will leave before then. Yeah, that's okay. So I will leave. Let us see. So let us see something from aquatic habitat. The method used for aquatic habitat depends on the organisms of interest. But generally, one common method, who can remind us of one common method that is used in one simple and common method that is used in aquatic sampling? Somebody remind us. You did that in general ecology. You did it. And even today, I talked briefly about it when I started this class. Who can remind us? I mean, if you answer my question, I may decide to mark your name and then give you some more marks as part of the continuous assessment. From what we did in the past in ecology, what is that method that is commonly used for study in aquatic habitat rather?
Good morning, sir.
Good morning, sir.
Alabi, Joshua Alabi.
Yes, you can say that from what we did in the past in ecology, not every one of us offers. So I might not be able to answer the question.
Sorry, I didn't.
And
The audio from your end is not very clear. Can you take it again? Maybe because of your internet.
Hello, sir. Can you hear me, sir?
Is that Omotayo Precious? Okay, Omotayo Precious. Let's start the top.
Yes, sir. So what I was trying to say is that many of us actually from microbiology haven't done general ecology. So most of the things you're referring to were not being taught to us.
Okay, somebody is facing an issue. There are people from microbiology that that they didn't do general ecology in the previous studies, and that most of the things I was referring to. No, I that 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 use of most is not appropriate. That's okay. The point I referred you to before now, before now, the the point I referred you to general ecology was from the data that are collected from the contract, how you can use it to calculate frequency, density, population size. That was the point that I referred you to general ecology before now. That was the point. That was the point. You have not missed anything apart from how to calculate frequency, density, and total number of the species from the data you have collected from the contract. Otherwise, you have not missed anything.
All right. Thank you very
Everything
much,
I've
sir.
taught is also common to other students from education, from CBG, from botany. That was the only point I referred you. And I just referred, even the equation itself, I copied it out. That's okay. So invariably,
Yes, sir. Yes, sir.
you have not missed anything. If I have not copied out the equations, you could now have a course to begin to go. But that I'll copy it out. That's okay. So I think the onus is on you now to go back and do some small reading. All that you need is what I've put out. Is that okay?
Yes, sir.
Yes, sir.
For microbiology. All that you need are those things I put out for you. All that you need are those things I put out. Now what I was going to talk about, they also did it in general ecology. And that was the question I was trying to ask, that what method we usually use to sample aquatic habitat. So that is again what it, even at that, I will still explain it all over. What we need, I will still explain it. I just wanted to know whether the other students can still remember what they learned from general ecology. So you have not missed anything, please. All that you need is what I'm giving you. Are you okay? And I will still give you.
Thank you.
Yes, sir. Thank you. Thank you very much, sir.
Thank you, sir.
To answer the question,
Okay, please ask a question.
no, I said to answer your question, the question you asked, the method used for. So, does grab sampling and water bottle sampling?
Sorry, wait. This person talking. What department are you from? What department are you from?
Microbiology, sir.
I know you are a microbiology person. Okay, thank you. No. What we need to talk about here is capture the capture method. Of course, these things you also did them in O-level biology. Is it a lie? Capture the capture method. That time you use that formula. You use A times B divided by C. You forgotten? You also did it. It's just the same thing we are iterating at this point. That's okay. I will talk about this now. Okay, let's go ahead. So, in aquatic sampling, one method that is generally used is capture. No, sorry, before then. Before then. Now, the method to be used depends on the organisms. That's for aquatic organisms. So, if the organisms under study are planktons or planktonic organisms, what are planktons? Yes, somebody remind us. What are planktons? Planktons are small floating organisms in aquatic habitat. You know, small floating organisms in aquatic habitat. You know, float organisms, live organisms, small floating live organisms in aquatic habitat. Broadly, there are two types. Some are plant-like, some are plant-like, while some are animal-like. What do we call plant-like plankton? We call them
Phytoplankton.
Good. What do we call animal-like plankton? We call them zooplankton. So, if the organisms under study are plankton, what do we use? We simply use a plankton net. The plankton net, they look somehow like insect net, but the mesh is smaller, you know, fine mesh. So that when you dip it in water, the planktons are caught in the net and the water content drains out. And then you can begin to look at what you have caught. You may fit a flow meter to determine the volume of water that is filtered out of the net. So the size of the mesh is determined by the organism of interest. But generally, it should be large enough to allow water to drain out. If it is not large enough to allow water to drain out, it will be a problem. That's okay. So to reduce clogging, it's good to make the net open so that water can drain out. So that is for plankton net. Now, this is a plankton net, like I said, it's looking like what do you call it? Insect net, but the mesh is much more smaller to collect microscopic organisms. You know, the microbiology people are used to this. So that's for that. Now, if the organisms is like fish or frog or crab, aquatic crab, you know, don't forget crab comes in different types in different species. Some are purely aquatic, some are semi-aquatic. Some are terrestrial. They are not going to that. They are looking at the aquatic ones. So, of course, like organisms like fish or something like a frog, we cannot be talking of plankton net. That's okay. So, what can we use? We can use there are many ways of sampling. There are many ways of sampling fish for ecological studies. You can use a fish net. You can use a gill net. That's fine. So this is a fish net. This is a fish net. This is a gill net. Now you can say this is a simple gill net. This man, what this man is doing, you know it. This man is setting the gill here. After I set it up, he will now leave. You understand? During high tide, water will overflow. That's okay. And this net will be covered up. When the tide recedes, when the tide goes back, some organisms are caught inside this gill. So that's the idea of a gill net. Is that okay? So the net is covered by tide. Okay, yes. And when high tide comes, so and then when the gauge recedes, when the water recedes, go back, then some unlucky organisms or fish is served. So that's one way. So the first was the fish net. The second was the gill net. Now, some animals are at the bottom. At the bottom of the pond or stream or even ocean. In such a case, you can use a kick sampler. When we say a kick sampler, it's something that you kick. So you kick it. As you kick it, it sort of disrupts the bottom of the pond and make the bottom organisms to float into water before it settles. You can see these people. This is a gig. This is what they call it now. This is a kick sampler. They dip it. You understand? They disrupt to the bottom, and then they pull it out. And then the bottom dwelling organisms that is caught, is now estimated. So for aquatic organisms, you may use, depending upon the organisms, you may use plankton net. You may use fish net. You may use fish net. You may use gill net. You may use kick sampler. You may use kick samplers. And then, yes, you are still talking about the same thing. You are still talking about the fish. You can also use what we call core samplers. Core samplers are used to collect samples of soil or soil sediment. So let us see each of these ones. This one is more sophisticated. It's used in the sea. This benthic grab is used in the sea. This one we are looking at has a long rope. You can see this is heavy metal that can go and sink to the deep of the sea. That's the basic graph. I mean graph. This is a core sampler. These are core samplers. All these could be deployed for artistic sampling. You don't need more than this. So this is where we are going to. So another thing to do when you want to study, one popular method that is used is capture the factor method. So at that point, we will do a copy for today's class. Next class, we will start from capture the factor method. So questions. So hopefully by next class, are you still with me? If you are with me, let me hear you talk.
Yes, sir.
Yeah, with you, sir.
Yes,
sir.
sir.
So we are going to draw the curtain for today's class.
Yes, sir. I'm with you, sir.
Yeah.
Okay. So let me hear you ask your questions if you have one. If there are no questions, we'll call it a day here. What should we do? Should we call it a day?
Yes, sir.
Yes, sir.
Okay.
Sorry,
So
sir, excuse me, sir.
okay.
Sir, please, will you share your slide?
This one. I will share the recording. I will send it to the class rep to send to you. Not only the
So what
slide.
about the slide, sir?
But the slide is contained. That's the slide that I'll be teaching with everything in the recording. And what else do you want? What's your Debra? Should I not? Okay, are you saying I should not share the recording?
Sir, share the recording too, sir.
Yes, the recording is okay, sir.
In fact, you have
Yes,
much.
sir. You can share the recording and the
We
slide,
need the
sir.
explanation. The explanation is vital, sir.
The recording contains all the slides now. I will not pull it out. That is fine. You even have the.
Okay, sir.
You even have you even have the YouTube link. That's a record of. Thank you very much. When do we meet next?
Thank you, sir.
Same time next week we will meet. Hopefully, I'll finish my part by then. So other people may want to come in if they are not free, so that you have time to do other things. That's okay. So that you have time to do other things. So read well, prepare well, and God will help you. So bye.
Bye, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir. Bye-bye.
Thank you. Bye bye. Thank you. Thank you.